Hello, everyone. This is Michael Naughton, and just a little information for you. This is how I've laid out my flipped classrooms in my high school health classes. So I do feel most comfortable with just speaking and having the information brought to you on the actual screen. Normally, probably you're used to seeing somebody video record themselves and actually seeing their person, but this is how I feel most comfortable. So just letting you know that. Anyways, our demo today is going to be a lab that is designed for the high school biology classes, and we'll be working in the general education inclusion class. This would be our big idea would be how our birth parents play a major role in our physical traits, which would be looking at the larger unit of human reproduction. In previous lessons, students have background information about genetics, about homozygous versus heterozygous, dominant and recessive genes, alleles, chromosomes, genotypes, phenotypes. We would be pretty much familiar with all of those vocabulary words, what they mean, and basically how to find out, what physical traits are passed from mother and father traditionally, or from birth parents to children. So here is how our lab would be going down. So before we would start, we would review basic concepts and vocabulary, like all the vocabulary words that I talked about, that we discussed, just making sure that the students know and remember that's what we're looking for and what they mean, especially when we're talking about dominant and recessive genes, homozygous and heterozygous alleles and chromosomes, genotypes and phenotypes. Now, a lot of those words are very close and very similar. So just making sure that they understand the subtle differences would play a big role in students' understanding and grasping of these concepts. So what we would do for the lab is we would pair up students. And then doesn't matter if they are male and female. Traditionally, looking at that would be the more traditional route of grabbing one male and one female to group them up and to say, okay, knowing that for human reproduction, for fertilization of an egg, we need a sperm from the male, which is the sex cell, and the egg from the female, which is their sex cell, and that's how fertilization would occur, and that's how a baby would be. It'd be in the process of being born or of being created. For this lab, we don't necessarily need a male and a female. We can have two males be pairs, two females be pairs, and we would just have one person be, in this case, the male, and the other person, in the case, be the female. And so, the question would be, what will you and your spouse's child look like based on your genetic traits and your spouse's genetic traits? So the procedure we would go over, well, the students, what they would need is they would need 3 beakers. They would need masking tape to label those 3 beakers. One would be the male beaker, one would be the female beaker, and one would be the baby beaker. The male and female beakers would be representative of the male sex cell and the female sex cell coming together to form the baby. They would also need some scissors, and those are basically only really the only equipment that would be used, other than the tables and the papers that are going to be handed out for the procedure to being used. In table 1, which we have right here, would be to find out each person's individual genotype. Their personal genotype, where they would be finding out what their hair color is, their eye color, etc., and they would be writing it out based on the description that they know of themselves, or the description that they could even use the partner to help tell them, what they have if they're unsure of certain things, like widow's peak or tongue roller, or even earlobes. So they, hopefully students would know that on their own, but at the same time, they're figuring out their own genotype, and then they're writing it out, and they're making sure that they're writing it out right here in each box. And so each person would have one of these tables. Then what they would do is they would transfer from this from table 1 over here to table 2. And so each person would be the chromosome 1 would be, the male in our group or the female in our group, and then chromosome 2 would be the other person. So then after they filled all of these out, what they would do is they would cut them up, and these are their alleles, which will be, creating the specific traits for their child. They would cut these out, and then they would put each one that they cut out into their respective male or female beakers. Okay. After that, what they will do is they will shake up those beakers and they will randomly select a trait or an allele, and they will look at it, and then they will write it down and copy it onto the genotype in table 3. If there is if a characteristic like the sex comes from the male beaker, then the characteristic from sex from the female beaker will not be used. Or based on who goes first with the random selection, they will select 6 different genotypes, and then the other person will select the other 6 different genotypes. So basically what we're looking for is 6 from the male and 6 from the female for 12 total traits to create the child. And then when they get those traits, what they're going to do is they're going to grab those 6 from the male, 6 from the female, shake them up, and put them into the baby beaker. Okay. And then what the next step that they're going to do is they are going to dump out the alleles from the baby beaker, and then they're going to pair them up for the genotype. So basically what they're going to do is they're just going to see which type of baby that is going to have it, whether they have a male or female, what hair color they're going to have, if there's a curl, eye color, all of these 12 descriptions that you can see right here on this slide. The next step that they would be taking is we would ask them to draw out a detailed picture of their child. And even though this picture doesn't have all of the traits and it's not the perfect picture, but what you can see is that there is labels, or there's at least arrows pointing to the labels describing or showing that there's a description of the different traits that the child was created. So we can see that there is blonde hair, and it is curly, the blue eyes, and then the freckles on the face. It would just 3 of the 12 examples of what we would be showing. After the drawing was completed, there is an analysis and conclusion questions. There's 5 questions that are determined based on prior knowledge before the lab and the knowledge that they have acquired from the lab as well, in determining the traits of 2 people when they are wanting to have a child, what their traits could potentially be in their child, or what their child would look like. Some differentiated or modified instruction that we have for this is that we did talk about a lot of vocabulary words. Those vocabulary words can be altered or simplified. The step-by-step instructions can be provided for completion of the lab, and as you can see with the tables are actually pretty much straightforward, where you can follow them, or someone can be assisted in following them and figuring out what can be picked. Also, students can be guided in determining their physical traits, where they can be helped out if they don't know what type of physical traits they're looking for. The number of traits can be reduced. The squares could also be pre-cut if we're dealing with people that cannot cut out squares on their own. And even drawing the final figure where the drawing is asked to be done, instead of a drawing, the description of the child can be verbally expressed in different, or it can just be expressed in different ways. So that is what I have for my science demo. I thank you for listening, and I look forward to watching the rest of the demos as they go.